Hello friends, I'm Irisa. I've just had 5 shots of espresso and I'm here with more biochemistry of toxins. Today's toxin is cholera toxin. Cholera is the disease that shaped mankind, or at least our plumbing system. It's caused by a toxin produced by this little bacterium called Vibrio cholerae. It's often spread through fecal contamination of a water source, and this is why I don't drink out of toilets, and you shouldn't either. The hallmark of cholera is diarrhea. Lots and lots and lots of diarrhea. Diarrhea like you've never imagined. It's painless, watery, non-bloody diarrhea that USMLE describes as rice water diarrhea. And there's a lot of it, up to 5 gallons a day. That's 5 gallons of straight-up diarrhea. That's like 10 of those 2-liter soda bottles. So it's not the cholera that kills you, it's the extreme dehydration from all that diarrhea that makes it so lethal. So let's forget about cholera toxin for a moment and talk about a transmembrane channel called CFTR. You've probably heard of this chloride channel before, but in the context of cystic fibrosis. This is a protein whose function is to let chloride ions pass from one side of a membrane to the other. And where negatively charged chloride goes, positively charged sodium follows. And where solutes go, water follows. So you're not just moving chloride, you're also dragging along sodium and water for the ride. That's a lot of important stuff we can't have spilling around willy-nilly, so CFTR has to be pretty tightly regulated. First, know that CFTR is only open when it's phosphorylated, like here. So let's look at this complicated machinery that controls when CFTR is phosphorylated, and therefore when it's open and letting those chloride ions spill out. Let's look at how this control system works on a normal day-to-day non-cholera, non-diarrhea basis. I want to draw your attention to the right side of the screen. Here, I've circled the G-protein coupled receptor in the membrane of the cell. Why is it called a G-protein coupled receptor? Well, because it has an associated heterotrimeric G-protein, which I've represented here with a hand in blue cuffs. When its ligand binds to the receptor, it causes a conformational change in its associated G-protein. The G-protein complex is made out of multiple subunits, but right now we only care about G-alpha, represented here by the hand holding a GDP. The conformation change causes G-alpha to separate from its beta and its gamma subunits, and for G-alpha to exchange GDP it's holding in exchange for GTP. And what do you know? We have an active G-alpha subunit bound to GTP that's ready to party. G-alpha has one function here, to turn on another protein called adenylyl cyclase, which I've chosen to represent in this diagram as a cute pink machine. So, adenylyl cyclase is essentially a machine that produces a load of cyclic AMP, or CAMP as I'm going to call it here, represented by these little gray ping pong balls, all dropping right into the cell. The basket on these seesaw represents intracellular CAMP levels, and before you know it, intracellular CAMP concentrations build up inside the cell, and high concentrations of cyclic AMP, CAMP, activate another protein called pKa, which is there on the left on the other side of the seesaw. Now pKa is the big doer here. pKa phosphorylates CFTR. And you know what happens with phosphorylated CFTR? It's open. Chloride ions can pass right through. Yay! Thank you, pKa! So let's look this over again. Here we have GPCR, ligand binding, causing a conformational change in its G-protein. That's your G-alpha subunit exchanging GDP for GTP. It's active, so it goes and it activates adenylyl cyclase, which makes CAMP. CAMP concentrations go up in the cell, which turns pKa on. pKa goes to phosphorylate CFTR. CFTR phosphorylated is open, and chloride ions go through. But now, let's throw cholera into the mix. Cholera toxin causes a conformational change in G-alpha that makes G-alpha unable to hydrolyze GTP to GDP to switch itself off. As a result, G-alpha is constantly turned on. But what does this mean for the rest of the control system? Well, a G-alpha that's on will be turning adenylyl cyclase on too. And it'll keep turning adenylyl cyclase on. Adenylyl cyclase being constantly on means CAMP is building up in the cell. Constantly high levels of CAMP in the cell means pKa is constantly on. pKa is always on, which means it's always phosphorylating CFTR, which means CFTR is open all the time and letting all the chloride out. And as we know, where chloride goes, sodium and water follow. All this ends up right in the intestinal lumen, and you can only imagine the resulting diarrhea. Now time for a fun fact. Did you know? Cholera might be the reason that cystic fibrosis still exists in the gene pool. You know how sickle cell trait provides protection against malaria? There's a theory that cystic fibrosis gene might provide protection against cholera, so the trait lives on via the heterozygotes. Why might this be? Think about what channel is mutated in cystic fibrosis. It's CFTR, the same one that cholera affects.